All right, ladies and gents. So in our last lecture video, we were talking about the light-dependent reactions. Again, the reactions that utilize the light energy in our photosystems to excite electrons to produce NADP and ATP through a concentration gradient of pumping hydrogen ions from the stroma into the thylakoid space. In this video, we're going to focus on the light-independent reactions. Again, those reactions that require the products of our light-dependent reactions to produce our high-energy sugars. So what's happening during these light-independent reactions? Well, during these reactions, we're going to be taking ATP and NADPH from the light-dependent reactions to produce high-energy sugars. So during the light-independent reactions, we call this the Calvin cycle. Again, it's named after the individual, sort of like the Krebs cycle. The Calvin cycle is the process of a series of chemical reactions that occur for us to produce these large sugar molecules. So NADPH and ATP, again, are very good at transmitting energy. The thing is that they're not very stable. So by using the Calvin cycle and utilizing these chemical reactions, cells can produce stable high energy molecules that they can use and store for a lot longer than they can do for ATP and NADPH. So let's go through the entire process here. So our whole goal in this is to produce a high energy sugar. And we know that sugars are just a combination of carbon, hydrogen, and oxygen. So what better to bring in other than a source of, so what's better than bringing in, say, carbon dioxide as a source of this carbon and oxygen, because it's very abundant in the air, and we can use this to actually funnel and fund the carbon debt that we need to produce these different molecules. So carbon dioxide is going to be absorbed by the cell via a particular structure on the outside of the leaf called the stoma. It's going to bring in CO2. This is the same place that we get rid of the O2 from our light-dependent reactions, and use that as a source of carbon for our reactions. So an enzyme in the stroma is going to combine these carbon dioxide molecules with a 5-carbon compound that's already present inside of the organelle. So the first reaction they have is we're using a molecule that we already know of, right, a 5-carbon sugar, or compound, not quite a sugar, to produce this 3-carbon compound, which again, if you think back to what's happening with cellular respiration, right, remember we take that glucose molecule and split it into a 3-carbon structure known as pyruvate or pyruvic acid. So here we're using essentially a similar thing, but we're kind of going backwards. It's like sort of the opposite reaction. Okay, so this 3-carbon compound that we're producing, and that's going to start being made inside the stroma. Right? Don't get stoma and stroma mixed up. They're very different structures, but the stroma is where this reaction is occurring. Okay. So for every 6 carbon dioxide molecules that enter the cycle, we can produce 12 3-carbon compounds. So we think about if we got 3 12-carbon compounds, we can end up producing in the end a single glucose. All right, and we'll get to that point here in the end. Okay. So as we're going through this process, other enzymes are going to react inside the chloroplast. Specifically, there's an enzyme called rubisco, which is one of the most prominent enzymes on the planet. It's the most common protein that you can find on Earth. And it's a protein that allows us to use this carbon dioxide to actually build these different sugars. It's an enzyme that's very important for the entire process. So other enzymes in the chloroplast are convert this. And again, we're using those high energy into higher energy forms using the ATP and the NADPH that we produced. So here you can see in this image essentially a basic breakdown of what we're going to be seeing in this process. So at mid-cycle, it's possible for the carbon compounds to split and not actually end up being made into glucose. So depending upon the needs of the cell, or like say we have like a low concentration of something that we need and the cell needs more of this particular product, they'll send signals that'll trigger the reaction to say like, okay, stop, we need more of this. Okay. So what's going to happen is at mid-cycle, 2 of these 12 3-carbon molecules are going to be removed, right? They're just going to be gone. And we're going to be using those 3-carbon molecules to build other things and other reactions. So say to produce other types of sugars that are used for structural components as opposed to energy. To build lipids, we might use them to go with amino acids to make some complex proteins or other compounds that the cell needs in order to function. So again, we're using 12 ATPs and we're using 12 NADPHs to produce these larger molecules as we go through. So the remaining 10 3-carbon molecules are going to be converted back into 6 5-carbon molecules to combine with 6 new carbon dioxide molecules to begin the next cycle. So again, this is where we're just going to add more CO2, we're going to have more reactions occur, and we're going to use more ATP from our light dependent reactions to help trigger this. So by taking those carbon strings, we're going to actually then convert them into a 5-carbon sugar. So in this process, the Calvin cycle is using 6 molecules of carbon dioxide to produce a single 6-carbon sugar molecule. So again, multiples of these are occurring at the same time. It's not just like one set of CO2 comes in and you have the reactions occur. I mean, because this is a complicated process of a bunch of different reactions that are happening in series, we're going to be producing a larger molecule from multiples of them. But then if we know that if we take 6 carbon dioxides, right, we need at least 6 carbons to produce a 6-carbon sugar. So we're using 6 carbon dioxide molecules to produce 1 sugar in this process. So the energies of these reactions, again, are supplied by the process products that we get from our light independent reactions. So again, the ADP, the NADPH, and ATP that we are using from those. So again, just a general summary. So the plant's going to use the sugars produced in the Calvin cycle to meet its energy needs. So it's going to do whatever it needs to do to produce enough sugar so that the cells can survive. And they're going to use whatever other products they're producing from this reaction, so other different carbon compounds, to help with growth and development, to build structural things like cellulose, like pectins, and those kinds of structures inside of plants that are necessary to create the overall structure and height of the particular creature. So when other organisms eat the plant, they're going to end up using these materials and using the energy that's stored here for their own purposes. So let's just summarize photosynthesis as a whole. So the two sets of photosynthetic reactions are going to work together. You have the light independent reactions and light dependent reactions. That the light independent reactions rely on the light dependent ones to give it the energy it needs for it to occur. Okay. So what's going to happen here is that by trapping the energy from the sun, we can convert it into a form of chemical energy that we can use. So chemical potential energy that we can use later. And these light independent reactions use this chemical energy that we make in the light dependent ones to produce stable, again, the keyword here is stable, high energy sugars, right? Ones that we can store for a long period of time. So again, ATP and ADP, as we do that conversion and make ATP, right, we've created a very high energy molecule. The problem is it's not very stable. It don't break down very readily, right? And that's why we use it in our cells to carry out reactions, because we can do them super fast. Okay? Right? Sugars requires a lot more to break them down. There's still a ton of energy stored inside of those, right? But again, we can store it, right? We can't store ATP. So in the process, animals, including humans, right, we are going to get our food and we're going to get our energy. We're going to get our food from the plants that are producing these carbon sugars, and we rely on these things that can photosynthesize for the oxygen we need to carry out cellular respiration. Okay. So again, what are some things that can affect photosynthesis as a process? And so the big 3 takeaways that we can talk about here are temperature, light intensity, and whether or not water is available. All 3 of these different things will affect the success of photosynthesis. So with photosynthesis as a reaction, it's really only going to function well in areas where the enzymes function well. And you may remember that enzymes, as there are proteins that are dependent upon the environment they are in. So if they fall outside of their homeostatic functions, right, the area in which they function the best, the enzyme's ability to convert light into chemical forms of energy goes down, right? It doesn't function as easily. So when the temperatures go above and below this range, right, the enzymes essentially stop working. So this is why, for example, during winter, you're going to see plants break down their photosynthetic pigments and store them for later, because the temperature outside is too cold for them to effectively carry out photosynthesis. Okay, so like in our angiosperms, that again, those plants that don't stay green year round, they'll do that. They're like, why am I trying? There's not enough light. The temperature's too low. The enzymes don't function very well. So let's just break it down, save it for when there's a time in the year that it can be done very effectively. Right? That's different than, say, like other groups like gymnosperms that have photosynthesis happening year round. Right? They have other modifications to their leaves that allow them to carry out the reaction. Again, still not as efficiently because it's cold, but still allow them to do it year round. Another thing that can affect the success of photosynthesis is the intensity of light. So if that plant is not receiving enough light energy, it's very possible that they're not able to produce as much sugars as necessary to carry out all of their other functions. So plants have adapted in various ways to deal with the amount of sunlight that is present in helping them produce the products that they need. So as you increase the intensity of light, you're going to increase the rate of photosynthesis, but it depends on the organism. Some aren't quite capable of taking advantage of this high intensity, and say they don't have enough of the pigment, or they're relying on other pigments because of where they are in the understory, so they're using other things to help speed the reaction along or to allow them to do that. And they can adjust their metabolism, slow it down, so they may not need to do photosynthesis as quickly. So this is why sometimes too, when you take like say a shade plant and put it in full sun, that it can get scorched, that it can end up damaging it. It's just because it doesn't have the ability to protect itself because it's not enough pigment to absorb that extra energy. Okay. But there is a point where we reach a maximum, right? There's just the rate of photosynthesis stops at a certain point, right? That it doesn't increase anymore and it stays pretty flat. And that just deals with the relative intensity of light. That if we reach like say an intensity of like 1,000 to even 1,200 micromoles of photons per meter squared per second, we're going to reach a point where we have saturation and the pigments just can't keep up. Then lastly is water. Since water is one of those things that we need as a raw material for photosynthesis, water is vital for the success of plants. That's why if you don't water houseplants, they end up dying. They can't photosynthesize anymore and they end up not being able to carry out the reactions that they need. So plants can live in dry conditions and they can live in areas where water is not very abundant, and they have different chemicals that they use to help speed the reaction along or to protect the cells from actual damage by allowing them to store water so they don't lose it through evaporation. So you might see leaves that are really thick and waxy. You might see leaves that have a very thick structure called a cuticle. You might see leaves that have other chemical adaptations for the photosynthetic reactions, or they have just different locations inside of the leaf where the photosynthesis will occur. So in order to store water and make plants more effective, what you'll see sometimes is that they can have variations in where photosynthesis takes place in the cell. So we talked about the stoma, right? This opening inside of the underside of the leaf that's going to allow material to transport inside or out of the cell, in this case gases and excess water and stuff like that. So most plants are going to be under these hot conditions. They're going to actually emit carbon dioxide and absorb carbon dioxide through these openings in the leaves, as well as some of them will absorb water from the air for that reaction too. Right? So when the carbon dioxide levels are, and so when we're dealing with like doing photosynthesis in extreme conditions, right? If you're opening the leaves to bring in carbon dioxide, you're also having the chance of losing water. So if you're going to be losing water through these openings, you have to control when they're open or closed to reduce the amount of water loss as you pick up carbon dioxide. So other plants have adapted like changes based on their environment to help minimize water loss. So we have some different photosynthetic pathways that exist besides the one that we addressed. So the one that we talked about that is just happening inside the chloroplast, inside the cell at all times, we call that C3. Right? This is just like the most normal, common type, I wouldn't say normal, but common type of photosynthesis. There's another type called C4. So C4 plants have specialized their chemical pathway to allow them to capture very low levels of carbon dioxide and pass them to the Calvin cycle. And so these plants are generally going to be plants like grasses, right? Things that are going to put their carbon into a different type of molecule. Instead of that 5-carbon chain, they're going to use a 4-carbon atom chain for the first compound. That's going to cause the reaction to essentially change, which allows them to need a little bit less carbon dioxide compared to other plants. So things like corn, sugarcane, sorghum, these are all grasses. Those generally carry that particular type of reaction as we go. Another type that we'll have is CAM plants. So CAM plants are those that are going to incorporate an entirely different acid. And you'll see these in things like cacti, some succulents. But what they're going to do is they're going to incorporate carbon dioxide into organic acids during photosynthesis in a particular metabolism called CAM. So Grassulian acidic metabolism. So CAM plants emit air into their leaves only at night. So this is a very temporal kind of thing. They only let it in at night because this carbon dioxide can then get trapped and we can store it by reacting it with other things. So during the daytime when the leaves are very sealed up, right, to protect water loss because of the intense heat and light that they're experiencing, the compounds that they've stored the CO2 in can release that CO2, enabling the process of the light independent reactions to occur. Okay, so CAM plants will be things like even pineapple trees, cacti, ice plants. There's a whole bunch of other things that can do CAM. But again, just know that CAM and C4 are variations on the whole process, light independent and dependent. So C4 plants, they just have a different sugar that they use, so they require less CO2, versus CAM plants that will take in CO2 at night, store it during the day, so it can continue the cellular reactions necessary. All right, so that's it for photosynthesis. So if you have questions, let me know.